Hello. So today I am here to deliver lecture about the spicules in sponges. See the picture. This is a type of sponge used in Japan as a gift for the newly coupled marriage. It is called glass sponge and the specimen is Euplectelia oene. In this particular specimen you just see here, you just see here see here these are the thing which are spine like structures. These are spicules. So what is spicules? Spicules are small needle rod like structure with crystalline appearance. With crystalline appearance, that means they are looks like glassy appearance. It consists of simple spine originating from a point. So this is point, this is simple spine, and this is an axis on which deposition of calcium carbonate or hydrated silica occurs to make it spicules. So these are the spicules which help in various function it has, because as it is looks like here, that means it is a spine, it can protect itself from the predator. So first one is why spicules are present there, protective in function, and due to the presence of these spicules it becomes stiff. So it also gives the appearance of stiffness, and ultimately as it is present it supports the total skeletal framework of the body. We have skeleton, but in case of sponge, skeleton is absent, there is present spicules. So again, due to the presence of these various types of spicules, we can identify various types of species. Now, how spicules are classified? Spicules are classified on the basis of size and on the basis of chemical composition. On the basis of size, spicules, when it is larger in size, it is called megasclera. When it is shorter in size, it is called microscleras. And at the same time, due to the deposition of organic material, such as when it is deposited by calcium carbonate, it is called calcareous, and when it is deposited by hydrated silica material, it is called siliceous. Now, what is megasclera? As the name indicate, the size will become increase in size, and the size, what is the size? 60 to 2000 micrometers, and it is a larger skeletal structure which help in the skeletal formation, and it is present as the sponge body is made up of two layers, outer and inner. Within this middle layer is called mesenchyme. So spicules are present there, and which help in the formation of skeletal structure of the body. So there are 5 types of megascleras are there: monaxon, triaxon, tetraxon, polyaxon, and spheres. First one is monaxon. Monaxon is a simple single axis point is there, and the deposition of chemical such as calcium carbonate or silica occurs there. So this monaxon can be also called as style. monaxon can be also called as style. So when in the monaxon, when the X is in one way, it is called monactinate monaxon. When it occurs in both way, it is called diactinal monaxon. So when this style has broad end, it is called knobbed end, it is called tylostyle, and when it is thorny process, it is called acanthostyle. And the main example of monaxon spicules present in the species Leucosolenia. Now triaxon. In that case, three spicules crossing each other at right angles with the formation of 6 hook-like structure. This is called triaxon. And various modification of occurs such that if the reduction due to the reduction of rays, and sometimes it is by branching of from monaxon, and that again it is by developing of spine. So this is called hexactinellida. That means example of triaxon is hexactinellida. In case of tetraxon, what does it mean? Four, see here, 4 tetraxons. So in that case, when all the 4 rays are similar in type, it is called calthrops. When it is one is elongated, it is called rhabdosome. It is called rhabdome, and other 3 which are crowned, they are called trions. So by loss of smaller rate, it may be called, if it is lost, then it is called dian. So if in case of tetraxon, if the spicules have both hand have disc, this is called amphidisc, and the example is called demospongy. Now it is called polyaxon, in which multiple equal rays arise from a simple point. It is called polyaxon, and it looks like a glassy in structure. An example is glass sponges. Now it is spheres in which spherical shapes, here spicules are spherical shaped, arranged in a circular way, then it is called spheres. And example is Euplectyleone, which have first shown in the first slide. Next is desmo. Desmo, in which silicons, whether it may be monaxon or maybe triaxon or maybe tetraxon, like this, they present in the center and outer side deposition of calcium carbonate or silica occurs, and it is found in demospongy. Now it is microscleres. In case of megascleres, the spicules are larger in size, but in case of here, spicules are smaller in size, so they can't help in the formation of particular skeleton, and they are dispersed in nature, and they are also shorter in size, about 10 to 60 micrometer, and they are of 2 types, spires and asters. So in case of spires, there are 4 types. Spire means it is spiral shaped. When it is this spiral is C shaped, it is called sigma. When it is bow shaped, it is called toxas. When the spicules in recurved hooks, it is called cheli, and sigma spire, it is spirally twisted. Now it is asters. They are of 2 types, small centers with long rays and large centers with small rays. These are the 2 types of asters. Again, small enterforms are of 3 types. oxyester, that which is a pointed rays, strongylester with rounded rays, and tilester with knobbed rays. Again, large center forms are spirester. Large centers form, this is large centers form with equal size, which is called spirester, and sterester with small just projected rays, these are called spirester. Again, other esters are like this. Stiptos are short, spiny, microsclear. Then cnidosters, it is rod shaped. Then pliosters with a few spine from a short section, it is few spine. Again, amphiesters, amphi means that means on the both sides they have disc-like structure. Again, microscopic forms of diactins are of three types: microbes, microaxia, and microstrongyle. Now, how spicule is developed? In case of, as I have already told you, that there are two outer layer and inner layer, and middle one is called mesenchyme. In the mesenchyme, a particular cell known as scleroblast they help in the formation of spicules. So, on the basis of developed spicules may be primary or secondary. So, how monaxon spicules are developed? See here, in the mesenchyme layers, these scleroblast divides, but the division of the scleroblast cell is incomplete, and at the same time both within between them, when the divide they move to each other, just in the repulsion case they move to here, and at the same time deposition of calcium carbonate occurs here, which ultimately forms the spicules. Similarly, at the triaxon also, triaxon means three scleroblast, and these three scleroblast form a triangular like structure, and of these three scleroblast they divide, and of the two outer one is called thickener, scleroblastal outer one is called thickener, and inner one is called founder. So all of these produce these spicule-like structures by the deposition of calcium carbonate. Similarly, at the all three layers, so ultimately form a triaxon. Same time with for the tetraxon formation, an additional, if it is triaxon, an additional scleroblast occurs there, and at the same time additional scleroblast occur there, and development of hexaclinal spicule and tetraxclinal spicules occur there. So this is all about formation of spicules in sponges. So if you like it, you just subscribe it. Thank you.